So the next step of our process is something called the TCA cycle. So sometimes we call it the TCA cycle, sometimes we call it the citric acid cycle, sometimes we'll call it the Krebs cycle. Those are all the same thing. Those are all just equivalent words. They basically name them after different molecules or different people involved with the discovery of this cycle, but the basic process is the same no matter what. Okay, so I'm just pulling up our view of the whole process of aerobic respiration. So we have out in the cytosol, outside the mitochondria, we have glycolysis happening. We're turning glucose into pyruvate. We took those pyruvate molecules, we funneled them into the center of the mitochondria. Then we broke pyruvate down into an acetyl group, carbon dioxide, some NADH. We ended up with a molecule called acetyl-CoA. And that's where we are now. So acetyl-CoA is going to feed here into step 3, into the citric acid cycle. So noting immediately here, so we made ATP during glycolysis. We did not make any ATP during the breakdown of pyruvate. So the breakdown of pyruvate is really just a linking step. Some people actually call it that, a linking step. And the point of breaking down pyruvate was really to get us into the citric acid cycle. We're going to make a little more ATP. So you can see here from the citric acid cycle, for each glucose molecule, we're going to end up with 2 more molecules of ATP. Bare bones of the citric acid cycle. So one thing is that we're in the mitochondrial matrix now. So keep an eye on where we are in the cell. Right? We're in the center of the mitochondria. Second thing to know about the citric acid cycle is that it is a metabolic cycle, which means it just keeps going in a loop. A cool thing about the citric acid cycle, like why we always draw it as a cycle, is that at the end of the citric acid cycle, we basically make the same molecule that we begin with, which is why we consider it a full loop. So some molecules are going into the cycle, other molecules are leaving the cycle, but it just keeps going and keeps making its own end product and looping back through. So we say that the molecule is regenerated. And specifically, the molecule that we end with is a molecule called oxaloacetate. Okay. So the first step of the citric acid cycle is that we basically feed in acetyl-CoA from our pyruvate oxidation into the cycle. So the cycle is going and going, and the acetyl-CoA is jumping in. Then we remove the acetyl group from acetyl-CoA. We tack it on to oxaloacetate, which is our beginning and end of the cycle. And we make citrate, which is also called citric acid, which is where the citric acid cycle gets its name from. Okay. The citric acid cycle is complex, and I'm actually not going to make you memorize all the steps. This is just so you understand why we call it a cycle. What I do want you to know is what the citric acid cycle makes, which is 2 molecules of CO2 for each loop of the citric acid cycle. So it's going to be 4 total molecules of CO2 for each molecule of glucose. For each round of the citric acid cycle, we also make 1 molecule of ATP. So for each glucose molecule, we have to do 2 cycles of the citric acid cycle. So we'll get 2 ATP total. The big point of the citric acid cycle, like its real purpose, honestly isn't to make ATP. Like we're already in the mitochondrial matrix. We already checked if there's oxygen. So we already know we're going to get to that final step where we have the big blast of 30 ATP molecules. So the real fundamental point of the citric acid cycle is to make a ton of electron carriers. So it's making a ton of electron carriers that we can next feed into our electron transport chain, which is how we're going to make a lot of energy. So we see that for every loop of the citric acid cycle, we make 3 molecules of NADH. which means that for each glucose molecule, we'd make 6 NADH. We also have another electron carrier that we haven't talked about. It's basically the same as NADH. We call it FADH2. It works the same. It's not like as important because we don't make as much of it, but consider it pretty much interchangeable with NADH. So it's another reduced electron carrier. We know it's reduced because it has those hydrogens on the end. Actually has 2 of them. We're going to rip off a hydrogen in the end of this process in order to help us make it. So here's a picture of the citric acid cycle. Again, don't worry, you don't have to memorize this, but I do want to show you what we're talking about. So here we see our acetyl-CoA coming in. The acetyl-CoA is jumping into a cycle that's pretty much already looping around, joining with oxalate acetate, forming citric. So that's the beginning of this process. And then as we go through the cycle, we create a molecule of NADH here and a molecule of CO2. So now we have fewer carbons. We make more NADH, more CO2, fewer carbons again. We make a little bit of ATP. We make a little bit of FADH2. At the end of the process, we make a little bit of NADH, and we go back to the beginning, get oxalate acetate again, keep looping around. Okay. So that's pretty much what you need to know about the cycle. The additional thing I want you to note here is that when we started cellular respiration, we had a glucose molecule. Glucose has 6 carbons. Of those 6 carbons, we just split them in half to make our 2 pyruvate molecules. So those were each 3 carbon molecules. Each of our pyruvate molecules lost a carbon during pyruvate oxidation in order to make these acetyl-CoA molecules. So the whole process was left with 4 total carbon molecules from that original glucose molecule, 2 in each of the 2 acetyl-CoA molecules. As we go through the citric acid cycle, we make 2 molecules of CO2. Both those CO2 molecules, the carbon in them, coming from that original glucose molecule. This means that by the end of the citric acid cycle, we've actually gotten rid of all the carbon from our original glucose. So it's all gone. We've breathed it all out. So here's the formal equation for our citric acid cycle. Put in 2 acetyl-CoA, we put in 2 NADH and 2 FADH, which is our precursor molecule for FADH2. We put those in so that we can make a ton of electron carriers, the main point of the citric acid cycle. So we get 6 NADH and 2 FADH2 out of this. We also finish up breathing out all our carbon from glucose, which is pretty cool. So we're getting rid of 4 carbon dioxide molecules. And we also made a little bit of ATP. So we got 2 ATP out of